All right, well, I don't think any discussion about relationship between genetics and behavior or genetics and neural outcomes would be complete without at least discussing briefly dementia and what we understand about that. Now, what I'll say up front is there are dozens and dozens of labs around the world working on various forms of dementia, various aspects of it. Little pieces. Our lab worked on it for 20 years in various ways, looking at very small pieces of things that actually led into some of the forms of dementia, particularly Alzheimer's. And we're trying, it's a complex problem, just like schizophrenia was a complex problem. Dementia itself, as you'll see, is a complex problem. But as we did before, let's just start out with a little bit of information about it. Alzheimer's, one of the primary causes of what we consider as dementia, is a growing issue, okay? The number of Americans that have experience with expression of dementia, as you can see from laser pointer here, from this chart over here, is increasing and increasing and expect to increase. We expect somewhere by 2050 there'll be something like 50 million cases around the world. It's one of the leading causes of death in America. COVID happens to be right up there right now, but if in normal times, Alzheimer's is a fairly common cause of morbidity. There are some estimates between 30% of seniors, particularly those over the age of 80, have some form of dementia. It may be minor, or it may be all the way to Alzheimer's and things. Once Alzheimer's is detected, either as the early form or the late form, the life expectancy is well less than a decade. So it is a high morbidity rate. As you can see from here, just sort of this figure over here is kind of the death rate per 1,000 around the world. North America, US, Australia, parts of Europe have very high rates right now. Other areas have lower rates due to Alzheimer's. Some of that has to do with how long people are living. Others have to do with environmental aspects. There are two primary forms. One is early onset, which has a fairly well-defined understanding of some of the genetics behind it. It's an autosomal dominant inheritance. There are at least 3 loci, maybe there's a 4th being discussed at the moment. It's familial, so it's inherited. This is an onset that can occur in the late 40s, early 50s, usually with a very short prognosis time to the morbidity outcome. And then the other type, the majority of Alzheimer's is much later. Its onset is on after in the 60s, 70s, 80s. This is the vast majority of it. It can be familial as well. It can be that there's if you look at pedigree, that there's multiple cases, but it's predominantly sporadic. We see individuals who have no family history of it or no indication, siblings don't have it, and then they'll have it. There are many loci thought to play a role, and there's other genetic risk factors. One of the big things about this is that within the next few years, we're expecting to see a dramatic increase in Alzheimer's throughout the United States, as we do in the world. These kind of the dark reds here, Georgia and other places, expecting to see why. Part of those is aging populations that are there. Certain areas have a younger population, others have a more aging population. Others happen to be health factors, individuals that food quality, medical access to medical facilities, that sort of things having an effect. All right, if we start looking at it overall, there is a large environmental component. We're talking about the sporadic form, the late onset form. Things like diet, chemical exposure, smoking, sleep apnea, stress, all sorts of things play a role. These are thought to play a role in 2 ways. One, by direct gene activation or inactivation. And there are a number of genes, ApoE, all sorts of things, P62, a number of genes that are thought to be related here. So one of them is just a direct interaction, an activation or deactivation in gene regions. The other is an epigenetic effect, particularly with some methylation of some HDACs and HATs forms. Now the genetics, once the genetics are there involved, and there is a propensity, again, it's familial, they can either have a direct phenotypic expression, so in other words, the genes become hyper-expressed, or in some cases knocked down. You'll see that there's it occurs, right? Another way that's interesting, and I think I just wanted to point it out here, is that there's mRNAs, microRNAs that do a job of splicing, as well as regulatory control of transcripts. They can be involved, and they can be modified through this, and then they will affect the phenotypic expression, and they can be affected by these epigenetic tags as well. So that's one of the areas. Whatever the cause, and again, it's a complex, it could be it's an interaction between all of these. You have this phenotypic expression, and that leads to the pathology, which is this progression. Neural systems start to fail, and then other systems fail, systematic nervous system, etc., start to fail. This pathology is complex. We think about it just as, well, people can't remember. They can't remember what happened yesterday or who people are, but they can remember these weird events from the past. They do things. That's all neurological relationships that go on, but there's lots of other things that are going on besides simply just neural connections that are forming. And this is sort of a diagram of what's happening here. We have lots of things that happened in Alzheimer's, and one of the reasons that you see various levels of it in forms. The things that are big happening here are things like mitochondrial dysfunction, ATP depletion, increased ROS. So once you've got reactive oxidative species, you've got all those other secondary mutational effects that are going on. There's modulation of gene expression. There's cellular stress in various ways, which leads to apoptosis, cell death. There's DNA damage, inflammation. There's all sorts of things that go on through here. One of the big outcomes, of course, is that we do see these amyloid plaques forming. Okay, these senile plaques, as they're called. They're basically, particularly in the neural systems, the cell is not processing as it should certain proteins, amyloid beta being one of them, amyloid plaques, and those are being, since it can't process them, it can't move them along the dynein pathway to be degraded in the lysosome. They're put along the outside, and they become aggregates, and they become just like a glue essentially on the outside of the cell or a paste, and neural systems can't fire between them. And so you end up with this neural cognitive decline as these build up over time. All right, and unfortunately, with some of the Alzheimer's, this can be fairly rapid outcome, because once the system starts to fail, it fails fairly quickly. As you can see, 4 to 8 years or so. Once the initial system is there, we get a combination of just death of neurons, as well as dysfunction, as well as then just those that are left are not highly functional. I just want to show you this, and there's this all these various forms that play a role here. Each, again, just as we've seen in most of these little diagrams, the greens are these proteins. So each one of these has a gene that's involved somewhere, and there's a case that it can be. One of the interesting things is that we really do not have a good definitive test for Alzheimer's. There's been some good progress in the last few years with some blood tests and things. What they're primarily looking at is this amyloid beta here, and the levels of those amyloid beta in the bloodstream, et cetera, as a custom. But amyloid beta is while it's clearly involved in this, it's not the only factor that's the primary form. So it's not a perfect diagnostic, although it's certainly interesting. Right now, as far as perfect or really definitive diagnostic tool for someone who's alive, whether they have Alzheimer's or not, there is none. Okay, there's cognitive tests you can give them. It's, you know, there's panels of questions and things and activities that you can, that are highly correlated and give you an idea that you have dementia in place. And then there's these new tests that are looking at things, but they're not, but we don't know. The only way you know is postmortem and that, you know, analysis of the brain structure and what's happened to the brain structure. Also, we don't have a good treatment, and we have some things that can help. A lot of the treatments are aimed at these amyloid systems and these aggregates, trying to either break them up or prevent them from being formed. But there's really no good treatment at the moment. Okay. All right, the other dementia, and again, there are many forms of dementia. Okay, and one's Alzheimer being one of the biggies because of the prevalence of it. One that's much less prevalent, but certainly has a pathway that in some ways is similar. And one I wanted to bring up because you studied it before in general genetics and other things, is Huntington's disease. Huntington's disease is a neurological gene, a fairly straightforward genetic product outcome that is a dominant. Okay, we consider it a dominant lethal, and only those individuals who tend to be heterozygous, having the Huntington's problem, you know, mutation, and then having a normal gene make it, and they generally make it into you know, their 40s or 50s, and then they die. By that time, of course, if they're going to reproduce, they probably have, so they may have passed it on. Now, the symptoms, there are lots of symptoms, okay? There's chorea. It used to be called Huntington's chorea, which is involuntary movements, kind of like Parkinson's, where they can't control, okay? They can't multitask, they're restless, they lose fine motor skills, they have trouble eventually swallowing and doing other things because, you know, sympathetic and other nervous system falls apart. There's a number of things that happen, balance problems. And you can see the brain, and this is very similar to what you would see if you did an image of an Alzheimer's brain. Okay, this is a normal side over here, all full of normal connections. And here we have, you know, a postmortem brain of someone that had Huntington's disease. The Huntington gene, one of the primary genes that's involved here, is found in chromosome 4, the P arm. And what's interesting about it is that it has a whole region of CAG repeats. Okay, CAG repeats, and you know, we know these are trinucleotide repeats. In a normal gene, it'll have anywhere from 20 to 36 or so, it says here, in the 30s of these repeats. When you get a problem is when you have more of these repeats, when you have 40, 45, 50. Again, you know, these trinucleotide repeats are often repeated due to slippage and other things. Excuse me. And there can be involved in things like Fragile X syndrome, right? One of the Fragile X is because we got repeats along the X chromosome. You get too many of those repeats and you end up with a breakage, and that can be familial, etc. Okay, so obviously these could be, if you got a lot of these repeats and they're passed on, you get this chain and you get a problem. Now, why Huntington's is, you know, causes so many problems, much like Alzheimer's, is that this HTT, each one of these red spots, is where the protein functions. It functions in things like adapting into dynein, right? We talked about dynein adapters. It's involved in mitochondrial function and transport into mitochondria and through the panels. It's due to export as well. And again, one of the things that we see that's in Huntington's is much like Alzheimer's, is abnormal secretion. So we get materials that can't be processed properly because they can't be moved along the dynein pathway, transport pathway. So they're pushed out on the outside of the cells. We get all sorts of other things that can happen to it. And again, you get a buildup, you get neurological damage, you get neural death. Obviously, you got massive neurological death here, apoptosis, which is, you know, cell death of the neural cells. They degrade. Once you have those degrading, you lose your ability to have you know, heart rate, feedback systems and all that functioning properly. So again, it's in the dementia family. Like Alzheimer's, there's Huntington's, there's PICS, which is another form of the dementia-like outcomes, and they're all have the point. They're again, once again, I keep pointing this out many times. These are all very complex systems, and many of them both relate into directly affecting DNA and how DNA expression occurs, which is you see over here, and others. Many of them have a mitochondrial input, causing mitochondrial function, and things like increased ROS triggers into mutational things that go on in DNA as well. So there's there's big, massive feedbacks and systems that work here. Okay. All right. Well, that's really kind of a brief look at dementia. Yeah, we could spend I keep saying this over and over and over again about these topics. We could spend a semester, we could spend your entire graduate career if you went into graduate school and much of your professional career just studying a small part of dementia because it's such a complex and important area. But that's at least an overview. A couple of those I do want to point out, things that you might not be familiar with and I think have some interesting outcomes are these next two, and we'll get to those in the next lectures.